I'm going to wander down here a little bit simply because you're all down here. So there's no escape. Okay. So that's been introduced, and I really want to talk about something that I'm particularly passionate about, which is astaxanthin. And I think probably, correct me if I'm wrong, a lot of people here probably have not heard a lot about astaxanthin and why it could possibly be important in your clinical practice. So I hope at the end of the day, at the end of this particular talk, you'll have a better appreciation of both what is astaxanthin and how you could apply it in your clinical practice. So the next slide. I would just go to me a little bit back. So just to give you some perspective, I am an Australian by birth, and my early academic training was at the University of Adelaide. I have a PhD in physiology and pharmacology. It's a little old, like myself. And 12 hours after I completed my doctorate, I moved to America, and I've spent my rest of my career in the USA. I've been a professor at a number of different medical schools. I was tenured as a full professor in my 30s. And I made a lot of sort of major contributions in diverse areas. So one of the ones that you may be interested in terms of the cardiovascular applications is back in the 80s, my team discovered that the endothelium, in terms of releasing a relaxing factor, which subsequently was found to be nitric oxide, is defective in cardiovascular disease. In fact, it's an early driver of things like hypertension. So that was the first observation, that the endothelial function is defective in cardiovascular disease. So a year after that discovery, they found out that the factor was nitric oxide. And Bob Furchgott, who was at a different New York medical school, won the Nobel Prize for that discovery. I moved on to do a lot of other nitric oxide stuff, particularly as it relates to inflammation. And I did a lot of different areas: gastroenterology, sepsis, infant nutrition, fetal development, arthritis, kidney, skin, and some neurology. So I've had, and stomach cancer. I had one of the highest rated grants in the history of the NIH for stomach cancer. So that's quite diverse. I'm going to try and focus on a nutritional product, a natural product, and its particular applications. So some elements are going to be a bit of a revision so that you understand where it applies and how it works in terms of not only pharmacology, but really some chemistry. So bear with me. I will make it a little simpler after I explain it. Next slide. So astaxanthin is a carotenoid. So what are other carotenoids? Beta-carotene, lycopene, lutein, zeaxanthin. So they're a little bit more common in your diet. Astaxanthin is less common. You really know it as more like coming from salmon. And here's the structure. It might be a little bit hard to see from the back. Very similar to beta-carotene here, sort of labeled in orange. And astaxanthin is very similar, except it's got these polar groups at both ends of the rings. They are essential for some of the unique characteristics of astaxanthin over beta-carotene. Okay? So that little bit of change, I'll let you know when to change it, is absolutely critical. So the other elements why you've probably never heard of it is that it's difficult to say. It's actually pronounced xanthin, right, for the X. And its primary source is an algae, also that has a rather difficult name to say, Haematococcus pluvialis. Just rolls off your tongue, doesn't it? Yes. There's some interesting elements to, hit the next slide, about this particular algae. It's a unicellular algae, a microalgae. So one of the things is, like all plants, it uses chlorophyll, right? It's an algae to create energy and substrates. And when it's stressed, it makes astaxanthin in large amounts. So it uses this carotenoid as its natural defense against stress, whether they be oxidative stress, osmotic stress, and it's its way to protect itself. And one of the things I always like to point out is that when nature is showing you the way in terms of protection, you need to pay attention because it's instructive and we could use that knowledge when applied effectively. Also, it concentrates in massive amounts, so krill is tiny amounts. This is really going up the food chain, right? And then you can go on to salmon and aspects like that, which we'll talk about later. Next one. So what are some of the really amazing features about astaxanthin? First of all, it is the most potent free radical scavenger. And here's some data looking at some other compounds as it relates to singlet oxygen, which is one specific free radical. So it's 110 times more than vitamin E, but some of these other antioxidants you'd be familiar with, green tea catechins, 560 times, 800 times more than CoQ10, which is really pertinent to this particular group for heart function, right? And if you go back down to vitamin C, it's 6,000 times more potent than vitamin C. It's very potent. Next slide. Okay, I want to review a little bit and explain what a free radical is. Most of the biomedical researchers, clinicians, healthcare providers really struggle with understanding the difference between an oxidant and a free radical. And I'm going to spend a little time explaining that because it is revealing as to why astaxanthin is so unique, so powerful, and so important. So one of the areas that I was researching in, as I mentioned, nitric oxide, I just want to point out some of the subtleties of different nitrogen oxides and what's a radical and what's not. This is just a tool. So explaining it here, these are not free radicals. Nitrous oxide, N2O, anesthetic, right? Nitronium cation, which is a nitrating substance, it's the thing that forms nitrosamines, which are carcinogens. Not nice. We don't want that, right? It's NOO plus. Nitrite. Again, one nitrogen, two oxygens, but that's negatively charged. That one's positively charged. And nitrite, as you know from being a way of preserving meat like bacon. No big deal. Nitric oxide is a free radical. And nitrogen dioxide is another free radical, so that's one and two oxygens. Very similar over here. This one's smog. It's the brown gas in smog. Again, this forms carcinogens, and this forms bacon. Very, very different actions. And nitric oxide, of course, is a vasodilator, neurotransmitter. The difference between what's a free radical and what is some other reactive species is that a free radical has an unpaired electron in its outer orbit. Nothing to do with charge, it's just spinning around unpaired. Why is that important? Because electrons love to spin in their orbits in pairs. The way I like to talk about it is dancing partners. Excellent. So, a free radical wants a dancing partner. Who here likes to go dancing by themselves? Right. If it was me, it would be particularly ugly. It would be probably a YouTube moment, but not very good. You need a dancing partner, right? So a free radical is either going to donate itself to another free electron to form a dancing partner or steal one. That's the nature of its reactivity. All it wants to do is orbit in a pair. And a very, very good free radical scavenger, by explanation, is the chemical that finds a dancing partner for that unpaired electron. And I'd like to consider that Astaxanthin is actually the tinder of free radical chemistry. For those of you who are familiar with social apps and singles, finding a dancing partner is important, and it's really the dating app of free radical chemistry. A lot of other so-called antioxidants are not really good dating apps. And the reason being is they may pair up that electron, but they stole the dancing partner from another situation where they were either married or in a significant relationship, and took the electron, made the dancing partner over here, but on this side, there's a very angry spouse. And that's why when you hear that sometimes antioxidants act as pro-oxidants, they did make the dancing partner, they paired it up here, but they've left the angry spouse. And you end up having as much damage, even more, than what you started with. So that's the sort of metaphors that I'd like to do. Let me give you some explanations of how it all works. Next one. So astaxanthin is a pure antioxidant. It has no appreciable pro-oxidant rate, but these other carotenoids do have both antioxidant and pro-oxidant characteristics. That means it will pair up that unpaired electron by stealing from a previous pair and making an angry spouse. Next slide. So what does all this mean as it relates to cell function? This is actually a study that's in human fibroblasts. It's a cell culture study, so here's the control. It's hit with singlet oxygen, a free radical, and you get this degree of cell death, right? These are all other so-called, antioxidants, what you would consider nice, friendly, effective free radical scavengers, and you'll note catechin, vitamin C, alpha-lipoic acid, the level of cell death is actually exacerbated over just singlet oxygen by itself. It's not helped the situation, it's made it worse. And it's the reason for it having a pro-oxidant activity. Tocopherol is not bad, CoQ10 is about normal, and astaxanthin is completely effective in blocking the cytotoxicity of singlet oxygen. It's a pure free radical scavenger and antioxidant. Next slide. So just taking that concept and moving it a little bit further, when you have oxidative stress, one of the things is that you change this tone within inside a cell that leads to an alteration in gene expression. There are a number of transcription factors which alter, they're like switches on genes. They either turn them off or on, specific genes. And that tone, that basic activity of oxidants inside cells determines which switches are on and which switches are off. And inflammation is particularly important in this particular area. NF-kappa B is the master switch for chronic inflammation, right? And NRF2 is often called the longevity switch. If you activate NRF2, you live longer, cells live longer. And heme oxygenase is just another downstream effect from NRF2. So this relative balance of oxidative stress affecting these switches, NF-kappa B or NRF2, determines what happens to a cell and what happens to inflammation, whether it maintains in a chronic mode or switches off. So oxidative stress is the driver, and astaxanthin is the modulator, right? Next slide. How do we know this? In terms of the studies, this is what happens in cell chemistry. So this is a particular type of cell. This is a neural cell. And what happens is NF-kappa B is normally anchored in the cytoplasm. It's got a physical attachment and it keeps it away from the DNA, so it can't switch on the genes. And when you hit it with an agonist, in this case lipopolysaccharide, it moves over and binds to the DNA, and you get this sort of reflection in the colors. When you have LPS, bacterial endotoxin, and astaxanthin, you can see that none of the NF-kappa B has moved over into the nucleus. It's not binding to the genes. It's not forming an epigenomic action, and the genes aren't turned on. So astaxanthin will negate chronic inflammation. And on the right, you can just see that these are reductions in interleukin-6 in two sources of glial cells relating to neurovascular inflammation. Okay, next slide. So moving on, understanding that oxidative stress is a source of chronic inflammation and astaxanthin is a preeminent free radical scavenger and negates the switches that regulate the chronicity of inflammation. How do we apply it and how can we use it? So it's working early on in terms of free radicals. We'll get to mitochondria in a second. It affects the switches and determines what's the degree of inflammation that's driving reductions in the quality of life. Astaxanthin will negate the accelerated aging that's associated with chronic inflammation and oxidative stress. So next slide. In addition, this sort of is a summary. So it will block direct free radicals, and these hit a lot of targets. Just mention a few here. Lipids are particularly important. And the reason being is that astaxanthin is soluble in lipids, right? And there's also evidence for DNA being damaged. You can regulate the genes. You can affect the mitochondria we'll talk about. And also it activates the endogenous systems that keep that stress down. There's a whole slew of actions that it's working to reduce that oxidative stress and all the sequelae to it from a carotenoid, from an algae. Next slide. One of the main sources of free radicals and dysfunction as it relates to aging is mitochondria. And so here we've got a slide showing that Time magazine sort of recognized that. I don't know if you know that Time often puts out an issue related to longevity. In this particular issue, they were not talking about longevity. They were talking about the quality of life with aging, which is really the health span, not the lifespan. So the health span is what portion of the lifespan are you healthy? And you want that to be as long as possible and really tight with that connection to the lifespan. You don't want to have half your life healthy and half your life unhealthy. You want that to be as coincident as possible. And one of the key elements is mitochondria. So next slide. So here we got a picture of a tissue. We have the mitochondria. It has lots of membranes, the cristae inside it. That's where we generate energy. And we know as we age that the mitochondria number decrease. They change their form. They swell. The number of membranes decrease. And what happens is those mitochondria are not producing ATP at the normal rate. ATP being the currency of energy for a cell. If you want to have any reaction, you're going to need some currency. So with aging, what happens is less money is being put in the bank, and you have the potential for a tissue or a cell to be energy bankrupt. The goal for healthy aging is not to go bankrupt with your cellular energy, right? So how do you do that? What I want to show you is astaxanthin can do that. So the other aspect is that this aging, defective mitochondria not only are not producing as much ATP, they're leaking free radicals. And so they're creating oxidative stress inside the cell and in the tissues around it, and that's leading to further dysfunction. So let's try and do a little review with an animation. I think that's the next one. Yeah. So what I'm going to do, Lee's going to hit the button for me. This is really the citric acid cycle feeding into the mitochondria membrane. This is what's happening to your substrate. There's CoQ10, electrons are bouncing around, there's protons going out into that space, and you end up converting ATP to ATP. But as a result, you're producing free radicals and protons. This can cause a lot of problems. Some of those free radicals can combine and make even further damage, right? You have natural defenses, you have superoxide dismutase, which will take superoxide down to water, and initially hydrogen peroxide, and then water as your defenses. Those defenses can be disrupted. If you get interaction with a superoxide and a metal, you form the hydroxyl radical, incredibly dangerous. First molecule it hits, it's just going to make a mess of it. Very reactive. That's how cells create the currency of energy. It's really fascinating as a mechanism, but it's prone to disruption. You need to protect it. The other thing is note that in this particular reaction, CoQ10 is associated early in that transfer of those electrons. So it's one of the reasons why CoQ10 is particularly important in creating energy and why it's useful in, say, states like heart failure. So next slide. So what happens with this system? This is the mitochondrial membrane sort of blowing out a little bit. You've got your mitochondrial DNA. Remember, that's different from the nuclear DNA. You get that from your mother. Thank you, Mum. Sorry, Dad. You're irrelevant. If you have these sort of leaks of free radicals, what happens? So just hit the slide. Hydroxyl radicals, just interacting with anything. Loves metals, makes a mess of that. Metal sulfhydryl groups, ruins them. And so with oxidative stress and dysfunction and leaky mitochondria, the whole system starts to fall apart. Next slide. So, just in summary, this is some of the actions of how astaxanthin could interact with that particular system. We know, and I didn't really get into it, but we know that carnitine is a sort of shuttle bus for fatty acids to go into mitochondria and burn them up for energy. It's not always glucose. Mitochondria will burn amino acids as well as fatty acids for energy. And that shuttle bus is turned on by astaxanthin. So if you need to use fatty acids for energy, astaxanthin is going to help. CoQ10 is part of the production system for the ATP, and astaxanthin protects that inner membrane. It allows CoQ10 to work a little bit better. Okay? It's protective of the membrane. And particularly as it relates to free radicals. And we'll explain that a little bit further on the next slide. So in essence, food, nutrients are going in, you're burning oxygen, it's going into the engine. The end result is carbon dioxide, possibly free radicals if they're not controlled, and you're getting ATP. So that's the engine, and astaxanthin is the oil that keeps the engine running smoothly. Okay? Let's show you a little bit more evidence related to the membranes. I talked about the membranes. Remember, astaxanthin is very lipophilic. And remember at the beginning I talked about the polar groups at the end of the molecule? By the way, all the bonds in the aliphatic chain in the center are in the trans position. It means the molecule's going straight out. If you have synthetic astaxanthin, it has these cis bonds at different locations. The whole thing is bent. What that negates is the ability of astaxanthin to form transmembrane bridges. So the polar groups bind to the protein because they're polar and charged. The lipid chain is out in the lipid bilayer and it's anchored on the other side. Any free radical that's swimming in that lipid layer is going to get picked up by astaxanthin, run down the chain, and then through a process of deprotonation, negates the free radical, finds its dancing partner, everyone's happy, no one's got an angry spouse. And it works so beautifully, it's like a fishing net. So if the free radicals are fish swimming in this river, This is a net that goes from one bank to another, and all the fish get captured. So not only is it potent, not only is it a pure antioxidant, pure free radical scavenger, it has the perfect structure to cross the membrane as a bridge, capture the free radicals, protect the lipids. Next slide. So in summary, in terms of some of the mechanisms for mitochondria, the other thing that we didn't discuss is it stimulates an enzyme called PGC1 alpha. That's an enzyme that's critical for mitochondrial biogenesis. What does that mean? You make more mitochondria. Some tissues have heavy burdens of energy, like the heart. If you don't make enough energy, it can't do its job. So make sure you make enough mitochondria, and that's one of the key enzymes that's involved. The other thing is we talked about transmembrane bridges, protecting membranes from lipid peroxidation. The non-technical term for lipid peroxidation is rancid. Yeah? Who likes to have rancid fats? Not really. It's not nice. How are you going to protect them? You need a pure free radical scavenger that's soluble in lipids. It turns on the transport protein for CPT1 for carnitine for fat utilization. And then if any free radicals do leak out, remember it maintains by not only killing those free radicals, it changes the epigenomic regulation of which genes are turned on or off. It's protecting at multiple layers mitochondrial function, and by that, the whole cell. Next slide. So that's some of the theory. Here's some of the practicality. Here we have some other cells, and in this case, the sort of precursor neuronal cells. So here is the control. This is the astaxanthin panel. In the middle, so what we're doing here is we've got a stain for mitochondria, fluorescent stain for mitochondria. And then we hit the cells with a toxin. There's two different types. We use 6-hydroxydopamine or DHA, fatty acid, that's been hit with a peroxide. So it's a free radical. It's a rancid form of fish oil. You've probably smelled some of those in your lifetime. What it will do to these particular cells is that it will kill these mitochondria and the cells. If you give astaxanthin, it's fully protected. Right? Whatever the poison was, 6-hydroxydopamine or a rancid lipid. In addition, on this slide, what it's showing is that the membrane potential of mitochondria, which is an index of how mitochondrial health is improved. So it's shifting this color spectrum to the red in these particular cells. It makes the mitochondria healthy. Next slide. So comparing it to something that you probably have heard of and utilize in your practice, which is CoQ10. Again, a reasonably well-known, heart-friendly, antioxidant free radical scavenger mitochondrial nutrient in terms of interacting with an electron transport chain. Both of them are free radical scavenger. Astaxanthin's 800 times more potent than CoQ10. Astaxanthin stimulates mitochondrial biogenesis. I don't know if CoQ10 does. It may be in the literature, but I haven't found it. CoQ10 is absolutely critical because it facilitates electron transport chain, but astaxanthin protects it. Slight difference. CoQ10 has no chance of forming a transmembrane bridge based on its structure. Astaxanthin is superb at that. Astaxanthin also works by shuttling carnitine. Astaxanthin also is now being known to cross the blood-brain barrier and also the blood-eye barrier for visual health. It improves membrane potential, reduces oxidative stress, both do that. Astaxanthin is superior, and improves blood lipid levels. Both of them do that, and we'll give some evidence in due course. Next. Okay, so Astaril has not done clinical trials with heart failure patients. It has historically focused on other areas. This is just an experience of one of the Japanese doctors. So We understand that it's a case study, limited in that regard, but I'm sure in your clinical experience, they're pretty remarkable findings. So let me describe some of the other studies, the clinical research that has been done by Astaril with its astaxanthin. And there are a number of factors that I think are impressive. So this one is looking at lipid peroxidation in overweight patients. Again, it's that oxidative damage to lipids. And I talked about mechanistically how powerful astaxanthin is. MDA is measuring a fragment of membrane lipids, and it goes down 35% by week 3 in this particular study with 5 milligrams a day. Total antioxidant capacity went up 22%, and the antioxidant superoxide dismutase also went up. I just want to spend a little bit of time on lipid peroxidation because to me it's very important and it's underappreciated. I think 2 weeks ago, there was a report that they've now put together a panel of 6 different indices of lipid peroxidation. And they showed that with cognitive decline leading to Alzheimer's, that the level of lipid peroxidation in the blood had an enormously high predictive rate on who went on to become an Alzheimer's patient, reflecting vascular inflammation and its particular role as an early driver of cognitive decline. So anything that lowers lipid peroxidation has a numerous benefits systemically. Next slide. Again, this is the same sort of scenario. Instead of being in obesity, now we have smokers. Yeah, that's going to create reactive oxygen species. Here we have a nice 32% reduction, again in MDA. Isoprostane is another indicator of membrane phospholipids. Actually, it comes from arachidonic acid as a 89% reduction by week 3 with astaxanthin. And again, on the defense side, superoxide dismutase went up 100%, and total antioxidant capacity was increased 69%. So again, that's an epigenomic faction activity raising SOD levels. Next slide. Looking at DNA damage, this is looking at this particular marker, 8-hydroxy-2-deoxyguanosine is a DNA fragment, gets cleaved if the DNA has been damaged by oxidants, and there was a nice 35% reduction with astaxanthin when given at 8 milligrams per day in a 4-week study. Next slide. Going to serum triglycerides, again, another lipid aspect, but one that you use quite commonly in terms of assessing cardiovascular risk. 25% reduction in 4 weeks in serum triglycerides, and in this particular study, at 6 milligrams or 12 milligrams, there was nice, significant dose-dependent decreases in serum triglycerides. Next slide. Serum lipid levels in women, looking at a profile with triglycerides, again, a nice reduction compared to the placebo and dose-dependent with given astaxanthin 2 or 8 milligrams. Next slide. Blood rheology. We have 2 slides, 2 studies that Astaril has done, and again, these are all Astaril studies on their ingredient. And so here we're looking at macular capillary blood flow, increase with astaxanthin 16%, no effect really in the placebo. There's a lot of evidence now that if you can improve macular blood flow, not only are you having an impact on macular degeneration, but also on glaucoma. Next slide. Switching from the macula to just muscle capillary blood flow, again, astaxanthin 6 milligrams a day over 4 weeks, increase blood flow in muscle 25%. So you get an improvement in sort of circulatory response. Yep. So again, related to potentially a diabetes situation where you may have either complications with obesity, cardiovascular disease, as part of metabolic syndrome. Astaxanthin has a role here with a reduction in HbA1c, not from a super high level, but certainly down. And tumor necrosis factor went down 30%. Tumor necrosis factor is a pro-inflammatory cytokine. It's epigenetically regulated, and astaxanthin reduced it profoundly. Next slide. Another one in postmenopausal women, as it relates to HbA1c, a nice reduction down to reasonable levels with 8 weeks of therapy. So getting back to the lipids again, this is an aspect that I'd like to sort of draw upon. So this is really just the pharmacokinetics of a single dose of astaxanthin, and you can see that it's monophasic. It has a Cmax at some time later. It's a slow rise, 11 hours. But one of the elements that I want to show in this particular graph is that because it's lipid soluble, it tracks with lipoproteins in plasma. Why is that important? Really, the main driver of vascular dysfunction, atherosclerosis, is whether the lipids that are delivered to blood vessels are oxidized or rancid. So if there are rancid lipids in the vascular wall, macrophages are going to come in and gobble them up because these lipids are rancid, they're dysfunctional. They then go on to form foam cells, which are absolutely the drivers of atherosclerosis. So it's the gobbling up of rancid lipids in the vascular wall that is a major driver of vascular dysfunction. If you can negate the delivery of rancid lipids and lipoproteins by astaxanthin because they're co-transported along with the lipids, you're going to have a big impact on vascular health. And that's really what this is alluding to. Next slide. So, how do you get it? By the way, that salmon, and I'm showing salmon here because salmon is one of the most common ways that we get astaxanthin in our diet. By the way, it has to be wild salmon, not farm salmon. Farm salmon uses synthetic astaxanthin. Remember, it has cis bonds, not trans. The whole thing is curled up. It's not as effective, it's not as well absorbed, and it's a bad free radical scavenger. Can't form a transmembrane bridge. Wild salmon has natural astaxanthin. I'm proud of that particular salmon that I caught. It's actually a king salmon, but king salmon don't have a lot of astaxanthin. In order to be the equivalent of 1 6 milligram soft gel of acetyl astaxanthin, you'd have to eat 12 servings of king salmon. I like it. That particular one was in a catch and release river. I had to give him back, which I didn't want to do. But I'd have to have 12 servings each day for 1 6 milligram capsule. Astaxanthin is a classic example of an absolutely brilliant natural product that you just can't get enough in your diet. And as a result, what you need to do is really supplement in order to take leverage of the actions of astaxanthin. Right? Some of the other forms of salmon are more potent or higher concentrations, coho salmon and sockeye. But if you were to do 12 milligrams, you're still, even with sockeye, having to eat 3 servings of salmon every day. Diet is not enough. It's a bit different if you're a seagull or something like that, where you're eating fish. That's the reason why seagulls' eyes are red. You know, they're getting it. Next slide. So, just a quick summary of Astaxanthin. There are different ways to grow the algae. You can have outdoor ponds. That's the least effective. There's a tubular system. And this is the Astaxanthin system, which is grown in Moses Lake, Washington State in the USA, and also in Stockholm, Sweden. They have these indoor photobioreactors where they've optimized the production, totally controlled. The reason why people are wearing these suits is not so that astaxanthin's dangerous. They're trying to protect the algae. It's a quality control that is a little different from having a pond just floating out in the air and anything fall into it. The other aspect is that as the algae gets stressed, it goes into this production system, and that stress is again indicative of the importance of astaxanthin in protecting cells. If it saves the algae, you can imagine what we'll do based on the evidence I've shown in a clinical setting. And also there's a whole other range of quality control. I was actually a customer of Astareals for a decade before I became a consultant. So I've actually seen the impact in our customers of astaxanthin from Astareal for a decade. So I have a lot of experience on the consumer point of view. So I think the last slide is that it? Oh, cost the locals. So again, I've just turned up from America. Apologies for not being around more often. But I'm passionate about this particular area. And this is an example of what you can get in Malaysia. And I would urge you to have continuing discussions with your team here at Medispec. And I really appreciate the opportunity to talk about one of my passionate areas, astaxanthin. They are your local team. They are the team that will look after you. And if you have any other questions, they will be able to supply them. Okay, thank you very much.